I think is a very exciting new approach to a number of different cancers and is now starting to be applied in multiple myeloma. I will say just up front that there are currently no real clinical outcomes data yet for CAR therapy in myeloma. So what I'm going to be doing is going through some of the rationale for this approach and some of the preclinical data, and particularly talking about some of the most promising targets. So I think there are a number of reasons why it might be reasonable to pursue an immune-targeted approach in multiple myeloma. I think first off is that despite the advances in survival that we're seeing with novel agents and autologous transplant, the vast majority of patients, unfortunately, are still not cured. It's also now been demonstrated in a number of labs that while there are clearly defects in T-cell and NK cell function in patients with myeloma, that if you can activate those cells appropriately ex vivo, you can get them to recognize and actually kill autologous myeloma cells. So if we're able to do the same thing in vivo, we might be able to overcome some of this dysfunction. There's an extensive experience now in the allogeneic setting demonstrating that T cells from donors can actually induce graft versus myeloma effect. Unfortunately, this is typically associated with graft versus host disease. And of course, the first immunotherapy, namely allogeneic transplant, can cure patients with myeloma. Unfortunately, we see very high mortality with full myeloablation. And what you can see are a number of studies like this that were performed quite a long time ago, where patients were getting fully myeloablative therapy. And you're getting, of course, these cohort of patients that are doing very well, and you get this actual hockey stick curve. But a lot of these studies are now no longer being done because of this very high treatment-related mortality associated with fully myeloablative conditioning. And so I think the challenge for us in terms of the field of cellular immunotherapy is how can we get this without this? And that's really led to a number of different approaches trying to modify T cells from patients themselves rather than from allogeneic donors where you can avoid the graft versus host disease. So first, a little bit of background about chimeric antigen receptors. This is a receptor that combines the recognition domain of an antibody with the signaling domain of a T cell. And so by various methods of gene transfer, you can actually stably express this chimeric antigen receptor on the T cell in addition to its endogenous TCR and thereby confer novel antigen specificity. And so basically what you have here is a signaling domain of a T cell receptor with a surface receptor that can now recognize molecules on the cell surface of tumor cells in the absence of any MHC restriction. In addition, there are now next generation CARs that add co-stimulatory domains such as 4-1BB or CD28, which have clearly been demonstrated to augment the proliferation and importantly the survival and persistence of these T cells. And this is really where we're starting to see some of the important and exciting clinical responses, particularly in some of the B-cell malignancies. So this is just an overview of how CAR T-cell therapy generally works. Patients undergo a leukapheresis to collect large amounts of peripheral blood mononuclear cells. Then these cells are activated to stimulate the T cells. At our center, we use antibody-coated beads targeting CD3 and CD28. This then allows the T cells to be transduced with the vector expressing the sequence for your chimeric antigen receptor. These are expanded in vitro. The beads are removed, and then these are infused back into the patient, often after some lymphodepleting chemotherapy, which can help facilitate their expansion in vivo. So I think the greatest success so far to date with CAR therapies have been the CD19 targeted CARs. You're going to be hearing a lot more about these tomorrow in the leukemia session, so I'm not going to go through this in detail. But basically, as a summary, there are now results from 8 different trials that have been published and over 27 trials ongoing and planned at different centers. This has been shown to be possible with both autologous and allogeneic T cells from donors. And what we've seen is responses in very heavily pretreated patients with B-cell malignancies, CLL, ALL, as well as non-Hodgkin's lymphomas. The response rates seem somewhat higher in ALL compared to CLL, again, relatively early data, and we don't quite know the reasons for this yet. And importantly, there have been some very durable complete remissions that have been seen over 3 years in some of our original CLL patients treated at Penn from just a single T-cell infusion. So really very powerful technology and a really new approach to treatment. Now there also are some unique toxicities associated with this. Tumor lysis syndrome and B-cell aplasia is expected and generally easily managed. What I think has been more difficult to manage, at least initially, has been this cytokine release syndrome. And this can be associated with very high fevers and rigors, leading to hemodynamic instability. Many of these patients end up in the intensive care unit with needing ventilatory support. You can get this HLH or macrophage activation syndrome that can cause neurologic dysfunction, seizures, obtundation. And this has been associated with very high levels of inflammatory cytokines, particularly IL-6. And some initial patients who develop this, you can abrogate this response with steroids, but this also seemed to kill off the T cells and the patient's clinical responses were lost. And what's been seen now in several patients is that giving an IL-6 receptor blocker, tocilizumab, can very rapidly abrogate this cytokine release syndrome and really improve this patient's outcomes. And so far, at least, we haven't seen a significant impact on the ability of the cells to mediate long-term responses. So I still think there's a lot to be learned about this, the optimal timing to when to use this. We currently try to wait until the patient is almost being wheeled to the ICU before we're doing this because we're trying not to abrogate the cells or the response too early, but I think this is something that really needs to be studied more. Unfortunately, the majority of patients have been able to respond to this treatment and recover. Now, I think there are a number of lessons that we can learn from prior CAR trials as we start to think about developing a CAR for myeloma. One thing that I think has become clear from several of the trials is that the expansion and actually persistence of these cells is very important. Some of the earliest CAR trials that lacked these co-stimulatory domains may have very transient responses, but then the cells were gone in a matter of weeks, and the responses didn't last. And what we're seeing now is more durable responses associated with these cells staying in the patient for months, to in some cases even years. There also was no clear dose-response relationship, at least that's been seen so far, and this sort of makes sense. This is a living cell that is going to expand in vivo, and so even giving smaller numbers will just expand up and be able to have an in vivo effect. I think another lesson is to be careful about immunogenicity of the construct itself. As these constructs are made, some of these antibodies were derived from murine sequences, and patients have developed human anti-mouse antibodies against these foreign sequences, and we actually had a patient in a mesothelin CAR trial that developed anaphylaxis after getting several doses of the CAR, fortunately recovering. And then I think another important lesson is to think very carefully about the target that you choose. And so it's ideal to try to pick a target that's really essential in the pathogenesis of the disease, or of that cell. For instance, we have seen an ALL patient who responded to CD19-directed CARs that then relapsed with CD19-negative disease. The other thing to think about is to beware of this concept of on-target, off-tumor toxicity. So there's a case report of a patient in a trial published a couple of years ago with a HER2 new targeted CAR who developed fatal pulmonary toxicity within 3 or 4 days after the infusion. And when they investigated further, they found low levels of HER2 new on normal lung tissue. And this, of course, was not expected or would have been predicted by the thousands of patients treated with trastuzumab or other HER2-targeted therapies. And so while we certainly use the experience of antibodies to help guide us in terms of what might be safe, it doesn't necessarily predict what's going to happen when you put in the T cells. So now moving on to trying to design a CAR for myeloma. I divided these candidate targets up into what I call the classics and the new models. So the classics are really those antigens that are been known to be expressed in myeloma cells for years, CD138 and 38, CD56, seen in about 80% of patients, and of course the light chain itself. And then there are a number of new models that are being developed in preclinical studies and some entering clinical trials. And I'm not going to have time to talk about all of these, but I'll highlight some of the ones that seem most promising. And before I get to that, I will mention one other potential target, and this is CD19. And this might not be something you would intuitively think of initially, since CD19 is a B-cell marker and typically not expressed on plasma cells, including malignant plasma cells. But we've known for a long time, actually, that myeloma patients harbor clonotypic cells within the B-cell compartment that have the same immunoglobulin gene rearrangements, the same light chain expression, in some cases the same chromosome abnormalities as the CD138 positive cells. And a number of studies have demonstrated that this CD19 positive CD138 negative fraction can actually recapitulate the myeloma, both in vitro as well as in vivo. And this is some data from some of Bill Matsui's papers from Johns Hopkins, where it's the CD38 negative fraction from the patient that actually causes the clonogenic growth. And you can actually block this myeloma colony formation with anti-CD19 antibodies. These CD19 positive 138 negative precursors tend to be more resistant to myeloma therapies. And actually Actually, we just heard yesterday from Keith Stewart about it, the recent paper, showing that bortezomib resistance can be associated with this downregulation of CD138 and XPP1S, and these were typically B-cell populations. Now, there is some conflicting data in this literature, so this by no means has been universally accepted, but overall I think there is enough data to generate this hypothesis that targeting CD19 positive cells following high-dose chemotherapy, which would be given to eliminate the mostly CD138 positive component, may eliminate these clonogenic precursors that contribute to relapse. And so this has led to a pilot study that's soon to open at our center of using the anti-CD19 CAR T cells following a salvage autologous stem cell transplant for patients with relapsed myeloma. We're particularly targeting patients who have relapsed within 12 months of an initial autologous transplant. This is typically a higher risk population that would be expected to have a short time to progression after a salvage transplant. And the thought process is, in addition to looking at safety and efficacy, we're going to compare the time to progression in each patient as their own internal control, and if the second TTP after the second transplant is longer than their first time to progression, that would provide some evidence that this may be actually doing something and would lead to a larger trial. Now, moving on to some other potential targets, CD138 and CD38 are classic targets expressed on the surface of majority of plasma cells. They're not solely expressed on plasma cells, however. CD138, for instance, is expressed on epithelial cells, particularly involving the gut. There is some antibody experience with an anti-CD38 immunoconjugate, conjugated to this maytansinoid, which did show some modest clinical activity, at least in a phase 1 trial, but there was some mucosal toxicity seen at higher doses as well. A CD138 CAR has been developed by a group from Beijing. It's a second-generation T-cell CAR and is now entered phase 1 trials, and we don't have any data available yet. This group also transduced NK cells. It's an NK92 cell line with the anti-CD138 CAR, and this also demonstrated activity, at least preclinically. And again, my concerns about this approach are the potential for mucosal toxicity, this on-target, off-tumor toxicity, as well as the fact that we know that CD138 can be downregulated in myeloma patients, particularly with advanced disease, and so this may be an easy way to avoid the immune response. CD38, we also have heard a lot about at this meeting, of course, in the setting of daratumumab and some of the other anti-CD38 monoclonal antibodies. CD38 is expressed in other immune cell subsets, including potentially a common myeloid progenitor. There is a CD38 CAR that has been developed by a group in Japan, and they have demonstrated activity in vitro as well as in xenograft models, in lymphoma, and now recently in a myeloma model as well. And again, the concerns because of the expression of this would be maybe more broad immune suppression or cytopenias, though fortunately we haven't seen that to date in the antibody experience. Now, the group at Baylor is taking an interesting approach, trying to actually target the light chain, and so kappa light chain is expressed in a little bit more than about 2/3 of myeloma patients. There also is an antibody against kappa light chain that's in phase 1 development. This is somewhat attractive in that there really is going to be very focused expression of this antigen. There shouldn't be any significant off-target toxicity, and potentially could be more immune suppressive because you're going to be leaving alone the lambda-restricted polyclonal B cells and plasma cells. There have been first and second generation CARs against kappa developed, and this trial actually has been open now for a year or two, using this kappa targeting CAR in patients with B-cell malignancies. And the very first data was just presented at the Tandem BMT meetings a few weeks ago. Very preliminary. So far, no dose-limiting toxicity. They actually have seen a little bit of activity in the non-Hodgkin's lymphoma patients. There was not much mention about the myeloma patients other than the disease still remains stable at this time. So we're awaiting further follow-up in this trial. And again, I think the concerns about this, number one, unlike some of the B-cell lymphomas, the light chain in myeloma is typically intracellular, so you're going to have a lot of surface expression the CAR is going to be able to recognize. And two, what is going to be the impact of very high levels of circulating free kappa light chains? Is this going to somehow cause a sink or be an issue for the ability of the cells to reach the target in the bone marrow? The next potential target I'll mention is another one we've heard about is CS1 or SLAM-F7. It's also known as CRAC and CD319, so there's a lot of different names for this molecule. This was originally described as an activating receptor on natural killer cells. It leads to NK cell activation. It actually binds through homotypic interactions with other CS1 expressing cells. And two papers in 2008 demonstrated that this was highly expressed on normal plasma cells as well as in malignant plasma cells in myeloma. And this is looking at mRNA expression from the patients treated at Arkansas. You can see there's a heterogeneity in the level of expression, but it was seen on almost all patients with myeloma. It's also expressed, however, on other immune cell subsets, including some activated T cells. And we've heard already about the antibody experience with elotuzumab, with really minimal toxicity, minimal single-agent activity, but synergistic activity, potentially with lenalidomide and dexamethasone, and phase 3 trials are ongoing. And despite the fact that this is expressed widely, there was not any sustained lymphopenia that was demonstrated, at least with the antibody. There's a transient decrease in lymphocytes as well as NK cell numbers on the first day, and then these numbers tend to return to normal. And with repeated dosing, the numbers stay up. So this does provide a little bit of reassurance about targeting this molecule. So the first CS1 CAR data that's been published is from the group at Ohio State. And they also took the approach of trying to transduce natural killer cell lines. So the NK92 and NKL are human cell lines. They demonstrated that they were able to transduce these cells to express the CS1 CAR, that this did enhance killing of CS1 expressing myeloma cell lines and did have some, albeit modest activity in a myeloma xenograft model. And I think this is a very interesting approach. These are potentially an off-the-shelf CAR, so rather than having to leukapheresis patients and spend weeks trying to generate autologous T cells or NK cells, this is a cell line that's available that you could transduce with your CAR and be ready to go in your patient. There's potentially less cytokine storm with this, and there's also this potential allo effect due to the cure mismatch that could be seen. I think, though, what we don't know yet is really, are these cells able to persist in vivo in patients? Are they able to expand? There's no particular, we expect less memory. Persistence of these cells, and there's also inhibitory receptors expressed on NK cells. So I think this is an interesting approach, but one that we really need more data about. And the same group, as well as other groups, are developing CS1 CAR T cells as well, and are demonstrating some activity, at least preclinically so far. And I think the last target that I'll talk about, and one of the ones that I personally think may be most promising, is BCMA, or B-cell maturation antigen. We also heard a little bit about this from Dr. Munshi earlier. This is a receptor for BAFF and APRIL. It's involved in normal B-cell homeostasis and maintaining B-cell and plasma cell survival. It is highly expressed in multiple myeloma cells, and importantly may play a role actually in the pathophysiology of myeloma cell survival. Signaling through BCMA promotes myeloma proliferation and survival and drug resistance. It also may be a natural target for the immune response in myeloma. There's a very interesting paper several years ago looking at two patients who achieved a CR after getting DLI for relapsed myeloma. And they screened the serum of these patients for antibodies against a number of antigens, and they found very high titers against BCMA, and found that these patients' serum was actually able to induce ADCC against autologous myeloma cells and cell lines. So this is a valid target. And we heard a little bit about the preclinical experience of this BCMA immunoconjugate. It's conjugated to an auristatin called MMAF, and these are data that is from Ken Anderson's lab. This can induce apoptosis as well as ADCC against myeloma cell lines. There is in vivo activity against myeloma xenografts. That's this curve here showing really excellent tumor eradication with the immunoconjugate, better than the naked antibody alone. And there's a phase 1 trial that's going to be opening in the next few months with this conjugate to give us our first experience targeting BCMA in humans. So the group at the NCI has published the first data looking at a BCMA CAR. These were transduced T cells. It was a second-generation CAR using the CD28 co-stimulatory domain. Importantly, they did a very detailed analysis of BCMA expression, both at mRNA and protein level in a whole host of normal tissues, and found expression only in B cells and plasma cells, again, somewhat reassuring about off-tumor toxicity. They were able to transduce T cells from both healthy donors as well as myeloma patients, and demonstrated activity against myeloma cell lines and primary autologous myeloma cells, and again showed the obligatory activity against xenograft models. So this hopefully will be entering into a clinical trial soon. This is something that we're also exploring at Penn in Carl June's group, and our CAR is utilizing the 4-1BB co-stimulatory molecule, but really seeing very similar activity. I'm not able to show that data yet. It's very preliminary, but in vitro as well as in vivo activity against myeloma cell lines. And so, again, we don't know what the effects of targeting BCMA in humans are going to be. We may have a little data from the immunoconjugate, but obviously we're going to need to do phase 1 trials in humans, and those are in the process of being designed. So to conclude, I think there are a number of promising targets for myeloma CAR therapy, and I didn't get a chance to talk about all of them, but there are even others that have preclinical data that look promising. We've shown that we can generate functional CAR T cells from myeloma patients despite the immune dysfunction in these patients. These cells have in vitro as well as in vivo activity in xenograft models, and clinical trials are underway. There's no efficacy data yet, but hopefully by this time next year we'll actually have a little bit of early data about whether there may be some efficacy with this approach. I think there's a lot of questions still about the optimal use of CARs in myeloma as well as other diseases. We're really still in the infancy of this. There's a lot of debate about the optimal co-stimulatory domains, the type of vector you use to transduce the cells, the optimal dose and schedule, as well as the need for lymphodepleting chemotherapy or not. So there's a lot of questions we have to answer, but I think a lot of promise here as well.